Welcome to the Biology Brief channel. Today we are going to refreshing the chapter Photosynthesis in Higher Plants or revising the chapter Photosynthesis in Higher Plants. This is a chapter from Plus 1 Biology, and you all know that this is an important chapter and many questions are usually asked for board exams and for NEET exam also. From 2020 NEET, 3 questions are being asked from this chapter. So we are going to discuss some questions asked in NEET examination from this chapter. First question is asked in 2024 NEET. Question: which of the following are required for light reaction? Required for light reaction in photosynthesis. Let us start from the right side. E option, light. This light reaction, light is very essential. Light is absorbed by photosystems. Photosystem, PS1, photosystems. Light is absorbed by photosystems with the help of chlorophyll pigment. So option D is also required for light reaction. Then when PS1 absorbs light, PS1 absorbs light and electron is ejected, that electron is used for the production of NADPH. The path of electron is completed there. Then another photosystem, that is for PS2, operates. It also excites electron, and this electron travels to PS1 for compensating the electron loss from PS1. This is the Z scheme of electron transport. And for compensating the electron lost from PS2, there takes place splitting up of water, and from that photolysis of water, electron is compensated in PS2. So for light reaction, water is also required. What about oxygen? Oxygen is the byproduct of photosynthesis. It is not the requirement, byproduct. Then carbon dioxide. Carbon dioxide is used for carboxylation during dark reaction. Carbon dioxide not required for light reaction. It is required for dark reaction for carboxylation. Dark reaction is also called a Calvin cycle. So carbon dioxide is also not required. Oxygen is also not required. Our answer for this question is option C, D and E, or option 2, C, D and E. The next question asked again in 2024. Which one of the following diffuse out of the chloroplast during photosynthesis? Direct question. We know that oxygen is the byproduct of photosynthesis and it diffuses out of the chloroplast. ADP and NADP, these are reactants for light reaction. ATP is one of the product of light reaction which is needed for dark reaction. Our answer for this question is option 3, oxygen. Next question asked in 2024 NEET exam. Observe the given figure. This is the figure. The figure is of Calvin cycle. Calvin cycle, you know, Calvin cycle. Identify the different stages labeled with alphabets A, B and C by selecting the correct option. Yes, ribulose 1,5-bisphosphate accepts carbon dioxide and produces 3-phosphoglycerate. Here carbon dioxide is added, or here there is carboxylation. Then 3-phosphoglycerate is reduced to form triose phosphate. There B stands for reduction. And triose phosphate, from triose phosphate hexosugar is formed from from a portion. The ribulose 1,5-bisphosphate is regenerated. So C stands for regeneration. We know that Calvin cycle consists of 3 stages: carboxylation, reduction and regeneration. A for carboxylation, B for reduction and C for regeneration. Option 3 is our answer for this question. Sometimes they label here A, B, C. in the place of the reactants, intermediary compounds. They give carboxylation reduction and regeneration and ask what are the labels A, B and C. You can easily answer that question also. What about fourth question? It is also asked in 2024. Which of the following are required for the dark reaction? Here the question is about the requirements of dark reaction. First option, light. Light is not needed for dark reaction. Chlorophyll. If there is no light, no pigment is needed. Chlorophyll also not needed. Carbon dioxide, it is needed. Carbon dioxide is assimilated during dark reaction. ATP and NADPH are reducing powers used up during dark reaction or Calvin cycle. So C, D, E are required. C, D and E. So option 3 will be our answer. Next question. Asked during 2024. How many molecules of ATP and NADPH are required for every molecule of carbon dioxide fixed in the Calvin cycle? Calvin cycle. So 1 molecule of carbon dioxide to be fixed. First stage, reduction and regeneration are there. For reduction, 2 ATP. For regeneration, 1 ATP are used up. For reduction, 2 NADPH is used. No NADPH is used during regeneration. So, in overall, for fixing 1 carbon dioxide molecule, 3 ATP molecules and 2 NADPH molecules are used up. So, for this question, 2 molecules of ATP, 3 molecules of ATP and 2 molecules of NADPH. Option 4 is our answer for this question. The next question. Asked during 2024. Statement type question. Here, statement 1. In C3 plants, some oxygen binds to rubisco. Hence, the carbon dioxide fixation is reduced. Here, rubisco binds with oxygen. Here takes place oxygenation. Oxygen takes place for photorespiration. Here the process will be photorespiration or C2 cycle. When rubisco combines with oxygen, oxygenation takes place. The process is photorespiration or C2 cycle. Here carbon dioxide fixation is reduced. The statement is true one, correct one. What about statement 2? In C4 plants, mesophyll cells show very little photorespiration while bundle sheath cells do not show photorespiration. Here in mesophyll cells, there is no rubisco. Rubisco is absent in mesophyll cells, so very little photorespiration. Even though oxygen is available, a very little photorespiration takes place because rubisco is absent there. In bundle sheath cells, rubisco is there, but carbon dioxide concentration is very high. So, no scope for oxygenation, only carboxylation takes place and do not show photorespiration. The statement is also correct. So, our answer is both statement 1 and 2 are true. Understand? Going to next question. 7th one. How many ATP and NADPH are required for the synthesis of 1 molecule of glucose during Calvin cycle? 2023 NEET exam. How many ATP and NADPH required for synthesis of 1 molecule of glucose? For 1 molecule of glucose, we need 6 carbon dioxide molecules to be assimilated. 6 carbon dioxide molecules for 1 molecule of glucose. So 1 molecule of carbon dioxide fixation, we need 3 ATP molecules and 2 NADPH. For fixing 1 molecule of carbon dioxide, we need 3 ATP and 2 NADPH. So fixing 6 carbon dioxide molecules or producing 1 molecule of glucose, 6 into 3, 18 ATP, and 6 into 2, 12 NADPH. So for synthesizing 1 molecule of glucose, we need 18 ATP and 12 NADPH. Fourth option is our answer. Fourth option. 18 ATP and 12 NADPH. Or coming to the next question, 8th question, 2023 NEET examination. Which micronutrient is required for splitting of water molecule during photosynthesis? Photolysis of water. The element or micronutrient required for photolysis of water during photosynthesis is manganese. Manganese. This is a direct question, easy question. Next one, 2023. The reaction center in PS2 has an absorption maxima at, we know that PS2, chlorophyll A is the reaction center and its absorption maximum is 680 nanometer. If it is PS1 instead of PS2, then PS1 also, for PS1 also, the reaction center is chlorophyll A and its absorption maximum is 700 nanometer. The answer will be changed. For the time being, the answer of this question is option 3, 680 nanometer. Next question, 2022 NEET examination. Which one of the following is not true regarding the release of energy during ATP synthesis through chemiosmosis? Chemiosmosis. Not true. We know that during photosynthesis, ATP synthesis takes place by chemiosmosis. Let us imagine the diagram. Here PS1, PS2. First option, reduction of NADP to NADPH on the stroma side of the membrane. We know that the reduction of NADP to NADPH takes place by PS1. Here NADPH is formed in the stroma side itself. So option 1 is a correct one, true one. Reduction of NADP to NADPH takes place in the stroma side by PS1. Breakdown of proton gradient. We know that here lumen, here stroma. In lumen, by this electron transport, H plus concentration is more. In stroma, H plus concentration will be less. Then there is a proton gradient. For chemiosmosis or to synthesize ATP, the proton gradient is broken down. The statement 2 is also true. Breakdown of electron gradient. There is no electron gradient in this case, so the option is not true. Fourth option, movement of protons across the membrane of stroma. Here the protons are moved back to stroma by facilitated diffusion. Here the movement is through FOF1 particle, and here ATP is also synthesized. You all know, you have studied. So option 4 is also correct one. So the not true option is option 3, breakdown of electron gradient. So option 3 will be answer to this question. Next question, asked in 2022. It is also a statement type question. In the primary carbon dioxide acceptance C for plants is phosphoenol pyruvate and is found in the mesophyll cells. In the case of C for plants, the primary carbon dioxide acceptor is PEP, phosphoenol pyruvate, and it is present in mesophyll cells. The statement is very correct. Statement 2. Mesophyll cells of C for plants lack the Rubisco enzyme. It is also correct. Mesophyll cells of C for plants lack Rubisco enzyme in order to minimize the oxygenation activity of Rubisco. No Rubisco enzyme in mesophyll cells, and the Rubisco enzymes are present in the bundle sheath cells, and in the case of C for plants, in bundle sheath cells, C3 cycle takes place starting from the action of Rubisco. So option 1 and 2 are both are correct in this question. So we select option 1 as our answer. Moving to next question asked in 2022. What is the role of large bundle sheath cells found around the vascular bundles in C4 plants? In the case of C4 plants. In C4 plants, we know that there is Cran's anatomy. Cran's a special anatomy called Cran's anatomy. There mesophyll cells are there. Mesophyll cells and bundle sheath cells. Bundle sheath cells. Prominent bundle sheath cells, ring-like bundle sheath cells. In mesophyll cells, you can see granule chloroplast. Granule chloroplast. In bundle sheaths, there is agranule chloroplast. In mesophyll cells, granule chloroplast, light is absorbed there. Here in bundle sheath cells, C3 cycle takes place, which is a dark reaction. No light is needed, so agranule chloroplast. Many agranule chloroplasts are there in bundle sheath cells. Here, please note down the word large bundle sheath cells. Why large bundle sheath cells are there? The answer. Check the options. 1, to protect the vascular bundles from high light intensity. It is not the answer. The protection of vascular bundles from light intensity is not the function of bundle sheath cells. To provide the site for photorespiratory pathway. Photorespiratory pathway is checked in C4 plants. So the option, the second option will not be the answer. To increase the number of chloroplasts for the operation of Calvin cycle. To increase the number of agranule chloroplasts for Calvin cycle, the large bundle sheaths are modified for that. For option 3 will be our answer. What about for option 4? To enable the plant to tolerate high temperature. This is also not the function of bundle sheath cells. So the answer to this question will be option 3, to increase the number of chloroplasts for operation of more Calvin cycles and increasing the productivity. What about next question? Asked in 2021 NEET examination. The first stable product of carbon dioxide fixation is sorghum. Sorghum. It's an indirect question. Sorghum is C4 plant. So the question can be read as the first stable product carbon dioxide fixation in C4 plants. What are the other examples for C4 plants? Sugarcane plant, sugarcane, maize, amaranthus. These are also C4 plants besides sorghum. The first stable, coming to the answer, a first stable product. Here carbon dioxide is combined with phosphoenol pyruvate with the help of the enzyme PEPcase, and the product is OAA, that is oxaloacetic acid. This is a direct question. Here the answer option 2 will be our answer for this question. What about the 14th question asked during 2021 NEET examination? Which one of the following statements is incorrect? Not that word in the question, incorrect. Both ATP and NADP are synthesized during non-cyclic photophosphorylation. Non-cyclic photophosphorylation. We know that PS1, when PS1 absorbs light, excites electron, there NADPH is formed and the passage of the pathway of electron is blocked. For another photosystem, for PS2, absorbing light, it also excites electron, and this electron travels to the PS1 to compensate the electron loss from that PS1 system. During this traveling of electron, here also ATP synthesized. The absence of electron in PS2 is compensated by photolysis of water. When this is Z scheme of electron transport, it is also called a non-cyclic photophosphorylation. During non-cyclic photophosphorylation, NADPH and ATP are synthesized, and the statement is a correct one. Second option, stroma lamellae have PS1 only and also lack NADPH reductase enzyme. Here PS1. In in stroma lamella only PS1 is there. PS1 is there. That P and NADP reductase enzyme is absent, so there is no formation of NADPH. When in stroma lamella PS1 absorbs light energy, electron is ejected, and that electron travels in a cyclic manner and returns to PS1 itself. It is called a cyclic photophosphorylation. During this, one molecule of ATP is synthesized. No synthesis of NADPH. So, option 2 is also correct one. In stroma lamella, PS2 is absent, only PS1 is there, and also lack NADPH reductase enzyme. Option 3, grana lamella have both PS1 and 2. Correct option. In grana lamella, non-cyclic photophosphorylation also takes place, and there PS1 and PS2 are present. Fourth option, cyclic photophosphorylation involves both PS1 and PS2. That option is a wrong one. We know that stroma lamella have PS1 only, and there is only cyclic photophosphorylation. For cyclic photophosphorylation, PS1 only needed. No need of PS2 and PS1. So, option 4 is a wrong one, and it is our answer for this question. We are moving to next question, which is asked in 2020 NEET examination. The oxygenation activity of Rubisco enzyme in photorespiration leads to the formation of Rubisco. The substrate is ribulose bisphosphate. Ribulose bisphosphate combines with oxygen. The products are phosphoglycerate and phosphoglycolate. Ribulose bisphosphate. Here the enzyme is Rubisco. Here oxygenation is the reaction. Here ribulose bisphosphate is a 5-carbon compound. Oxygen has no carbon. Phosphoglycerate is a 3-carbon compound. We know that in C3 plants, this is the first stable compound. The cycle is called C3 because phosphoglycerate is 3-carbon compound. The remaining 2 carbons are there, so phosphoglycolate is a 2-carbon compound. When Rubisco combines with oxygen, we will get 1 molecule of 3-carbon compound and 1 molecule of 2-carbon compound. 2 molecules of 3-carbon compound wrong option, 1 molecule of 3-carbon wrong, 1 molecule of 6-carbon compound wrong, 1 molecule of 3-carbon compound and 1 molecule of 2-carbon compound is our answer for this question. We can find the answer very easily. Yes. Next question, 16th question asked in 2020 NEET examination. In the light reaction, plastoquinone facilitates the transfer of electrons from. Here we have to remember the picture. Thylakoid membrane is there, stroma side, this is lumen. Here PS1, PS2. Here cytochrome B6F complex. Plastoquinone is placed here. Plastoquinone. It is a proton acceptor. It accepts proton from stroma, electron from PS2, and transfers electron to cytochrome B6, cytochrome F, and passes the protons to lumen side. In the light reaction, plastoquinone, what is the role of plastoquinone? First option, see the first option, PS2 to cytochrome B6. Here plastoquinone is placed between PS2 and cytochrome B6F. It absorbs proton from the stroma, electron from PS2, and transfers electron to cytochrome and F complex and proton to lumen. And answer for this question is, we can deduce from the first option itself. What about second option? Cytochrome B6F complex to PS1. That electron transfer from cytochrome B6F to PS1 is through the electron acceptor plastocyanin, which is present in the lumen side of that membrane. So for the answer of second option is plastocyanin. PS to NADH plus not the answer. Fourth option not the answer. It is clear. First option is our answer. You can easily find out if the picture is in your mind. Yes. What about next question asked during 2020 year? Which one of the following statement is incorrect? Find out the incorrect statement. Rubisco action. What is given here? Rubisco action. Rubisco action means C3 cycle requires ATP and NADPH. The reducing powers for carbon dioxide fixation or reducing power for C3 cycle is ATP and NADPH. So ATP and NADPH is needed for Rubisco action, and the option is correct. Rubisco is a bifunctional enzyme. Rubisco. The full form of Rubisco is ribulose bisphosphate carboxylase oxygenase. It's a full form of that enzyme, Rubisco. Ribulose bisphosphate carboxylase oxygenase. It undertakes carboxylation action and oxygenation action also. Here, when carboxylation action, if the concentration of carbon dioxide is more in the medium, carboxylation takes place. If the concentration of oxygen is more, oxygenation takes place. So the statement 2 is also correct one, because Rubisco is a bifunctional enzyme. It is both carboxylase and oxygenase. Third option, in C for plants, the site of Rubisco action is mesophyll cells. In C for plants, in mesophyll cells, Rubisco is absent. No Rubisco in mesophyll cells to check photorespiration or oxygenation action. There the enzyme is PEPKase. Primary carbon dioxide acceptor is PEPKase and not Rubisco in mesophyll cells, because Rubisco is absent in mesophyll cells, and the option 3 is a wrong one. Fourth option, the substrate molecule for Rubisco enzyme is in bundle sheath cells. By C4 pathway, more carbon dioxide concentration is bundle sheath cells. In bundle sheath cells, more carbon dioxide concentration by C4 pathway. So there is no scope for Rubisco. Go for oxygenation. Here Rubisco is compelled for carboxylase action. So the option 4 is also a correct one. So our answer for this question is third option. A third option is a wrong statement. The next question is 18th one. In Hatch and Slack pathway, the primary carbon dioxide acceptor is, we know that Hatch and Slack pathway is also called C4 pathway. C4 pathway. It is named after the discoverers of that pathway, Hatch and Slack pathway. The primary carbon dioxide acceptor, carbon dioxide primarily accepted by phosphoenol pyruvate to form oxaloacetic acid. PEP is a 3-carbon compound, carbon dioxide there is 1 carbon, so OAA is a 4-carbon compound. First stable product is OAA, which is a 4-carbon compound, so the name C4 pathway. Here the primary carbon dioxide acceptor is nothing but phosphoenol pyruvate. This is a direct question. Our answer is fourth option, phosphoenol pyruvate. Next question asked in 2018 NEET examination. Oxygen is not produced during photosynthesis by, there are two types of photosynthesis, anoxygenic and oxygenic. Here in green sulfur bacteria, there is Anoxygenic photosynthesis, where here oxygen is not evolved because here H2S is the hydrogen donor, not water. So there is no evolution of oxygen in green sulfur bacteria, and the answer to this question is green sulfur bacteria. Nostoc, which is a blue-green alga, Cara is a green alga, Cycas, higher plant, gymnosperm. In these plants, there is oxygenic photosynthesis. Oxygen is evolved during photosynthesis. So, the answer to our question is first option, green sulfur bacteria. This is the last question asked during 2017 NEET examination. It is a statement. There are some statements. The question is about factors affecting photosynthesis. Last portion of that chapter. Factors affecting photosynthesis. With reference to factors affecting photosynthesis, which one of the following statements is not correct? Carefully examine the statements. Increasing atmospheric carbon dioxide concentration up to 0.05% can enhance carbon dioxide fixation rate. We know that carbon dioxide, the major limiting factor of photosynthesis, carbon dioxide, because carbon dioxide is present in the atmosphere in 0.03% to 0.04%. Very little amount of carbon dioxide is present in atmosphere. So it is always a limiting factor for photosynthesis. But if the carbon dioxide concentration is increased up to 0.05%, there is enhancing of carbon dioxide fixation or photosynthetic rate. But beyond this limit, we are giving more carbon dioxide for a long period. The photosynthetic rate will be decreased. This statement is a correct one, as in the textbook. Second option. C3 plants respond to higher temperatures with enhanced photosynthesis. The statement is a wrong one. It is a character of C4 plants. C4 plants respond to higher temperature with enhanced photosynthesis, while C4 plants have much lower temperature optimum. Here C3 plants. C3 plants need much lower temperature optimum, whereas C4 plants are adapted to higher temperature with enhanced photosynthesis. The statement is a wrong one. The C3 and C4 are misplaced. What about third option? Tomato is a greenhouse crop which can be grown in carbon dioxide enriched atmosphere for higher yield. Let's see that. Carbon dioxide saturation point for C4 plants is 360 microliter per liter. A lower carbon dioxide saturation point is for C4 plants. In the case of C3 plants, much higher, 450 microliter per liter of atmospheric air. Much higher rate is for C3 plants. So, C3 plants show greater photosynthetic rate in higher carbon dioxide concentrations. Tomato is a C3 plant. If tomato is grown in greenhouse where carbon dioxide concentration much more than atmospheric, it gives higher yield. The statement is very correct. There is another plant given in the example, that is bell pepper or capsicum. Capsicum is also grown in greenhouses for increased higher yield due to this property. Fourth option, light saturation for carbon dioxide fixation occurs at 10% full sunlight. Light saturation for carbon dioxide fixation occurs at 10% full sunlight. In the textbook, it is noted that light is never a limiting factor. Light is never a limiting factor because when there is full sunlight and plants absorb 10%, it is enough for that plant for optimum photosynthesis because it is the point of light saturation. remaining 90%, the plants cannot absorb the remaining 90% because its limit is over. So the light saturation is 10% full sunlight for photosynthesis. Correct option. Here not correct or wrong option is asked in this question. So our answer will be second option. I think that you are enjoyed this revision session, and the session will be very useful.